Hi, my name is Parker Allen. I am a grad student at Colorado State University and one of the interns for BBF this past summer. And I'll be talking to y'all today about Ruppia maritima, uh, which is a seagrass found on the Black Sea coast and how it can be used for coastal conservation. So Ruppia maritima is found throughout Europe and North America in rivers, lakes, and streams. And though it's found on the Black Sea in brackish waters, woodland grass, which is the common name for Ruppia maritima, is really a salt-tolerant freshwater species. We worked in the lakes surrounding lakes and basins surrounding Burgas, Bulgaria, which are part of a major migratory bird route called the Via Pontica, which stretches from Eastern Europe into Southern Africa. Um, the Burgas Lakes will see over 400 species of birds throughout the migration, and woodland grass serves an important purpose as both habitat and food source for these bird species on the migration and for resident bird populations. Um, the grass functions as a breeding ground for brine shrimp, which is the main food source of flamingos in the area, along with other water bird species, and also works to stabilize the soils in the area, along with sequestering carbon for the area as well. So as you can see on our poster, we sort of identified optimal parameters for the species, and throughout the summer we worked on different techniques to transplant Ruppia maritima from one basin to ones that where no Ruppia maritima is established. And our objectives were finding the viability of differing methods for this small scale transplantation, developing a test scheme to improve the water quality through covering the viability of transplanting Ruppia maritima in the five basins, along with creating new habitat for macroinvertebrates and other waterfowl species. So we came up with two main options for transplantation. The turf method, which is already being used by BBF, involves taking a large wooden frame with compostable materials and pulling up woodland grass, putting it into bundles, and putting those bundles into the frame and submerging that frame into the substrate in a new basin. And then the sod method, which involves simply taking a block of substrate with Ruppia maritima attached and moving that entire block to a donation site. The turf method is great because you get a lot of plants into a very small area, and the viability of that one tray goes up pretty high because of how many plants are in it. But it also is very time intensive. And the sod method actually came about because we were finding ways to get rid of the frame, because the frame takes a long time to make as well. The sod method is great because the plants don't experience any trauma from being pulled out of the ground. They stay in the same substrate, and we can just move that block, that piece of sod with it in there. But they are heavier, and you move less plant material than you would with the frame method. Our main assessment for figuring out the status of our transplanted areas was with three indicators: bearing fruit, um, new vegetation buds, and new growth in general, and then those buds beginning to bloom. So far, we have seen that the Ruppia maritima has spread to other places that we've put it in, and so those indicators have worked as good benchmarks. We then also came up with future recommendations, which the first step is always to monitor and evaluate those transplanted areas to see signs of change, mainly in water chemistry and temperature, to then document those changes in water chemistry over time, and also to see if the establishment of Ruppia maritima in those basins changes the water chemistry for the better. Further research the ecosystem services, the benefits that Ruppia maritima gives people in the area, communicate those benefits to the public and explain to them why this area is very important to conserve. Do work and research into why previous populations died in the area, and then lastly, talking with other conservationists throughout the region and the continent to figure out new methods for transplantation and sharing successes and failures in doing so. Thanks so much, and we hope to hear any questions from y'all via email.